In this tutorial, you will learn how to generate 4 different kinds of heat maps using the Dataset Explorer tool in tranSMART. A heat map is a matrix of data points for a particular set of biomarkers, such as genes, at a particular point in time, and/or for a particular tissue sample in the study, as measured by each subject in the study. Heat maps for tranSMART can display data points for up to 1,000 samples. We will generate the following types of heat maps in tranSMART Dataset Explorer: standard heat maps, hierarchical clustering, k-means clustering, and marker selections. We are using the public instance of tranSMART available from transmartfoundation.org. When you have launched the public instance of tranSMART, click on the Dataset Explorer tab in the top menu bar to get started. A standard heat map is a visualization of biomarker data points with no indication of patterns, grouping, or differentiation among data points. Open the Public Studies folder to view the available studies you have to work with. In a standard heat map, you can select 1 or 2 subsets to examine. You can use an entire study for your one subset. Let's choose a breast cancer study by Sorley as our one subset. Click and drag the folder to the Subset 1 box. Click the Advanced Workflow tab. There's a button that says Analysis. Click to view a drop-down and select Heatmap. All heat maps require a heatmap variable defined by a high-dimensional data node. To find our high-dimensional data node, we will need to expand the breast cancer study by Sorley folder. Click to expand the main folder. We now see biomarker data and sample factors. Expand the biomarker data folder. Then expand the gene expression folder. Expand the Stanford cDNA microarrays folder. And finally, we see data labeled with a DNA symbol to the left. We know that we have found high-dimensional data when we see it is labeled with the DNA symbol. Click and drag the high-dimensional data to the heatmap variable definition box. Be sure to click the high-dimensional data button or your analysis will not work. When you click the button, a Compare Subsets Pathway Selection dialog box appears. Make selections in this pop-up that will meet your analysis needs. For this example, I am using the default selections. Click Apply Selections from the pop-up window. Now scroll down to the bottom of the page and click Run. The analysis will take a few moments. When the analysis is completed, the heat map will appear on screen. You will need to scroll down to view it. Click on the image of the heat map to open it in a new window for improved readability. Subsets appear on the top edge or x-axis. In this case, we only have one subset, so the entire bar is marked in orange. Biomarkers, such as genes and probes, appear on the right edge or y-axis. We are now seeing differential expression of the genes. Individual values in the matrix are depicted in colors. Higher than normal expression is depicted in red, and lower than normal expression is depicted in green. Back in the tranSMART window, you can download a PDF version of the standard heat map by clicking the Save to PDF button in the right-hand corner. In the instance of tranSMART, click on the Dataset Explorer tab in the top menu bar to get started. Hierarchical clustering allows us to visualize patterns of related data points in gene expression data. This time, let's use two subsets for comparison for the Hierarchical Clustering heat map. Expand the Public Studies folder, and we will still be using data from the breast cancer study by Sorley for this example. Expand the breast cancer study by Sorley folder, and expand the sample factors folder. Next, expand the T&M category folder and the tumor stages folder. I will click the T3 data and drag it into the subset 1 comparison box. For subset 2, I will expand the estrogen receptor array folder and drag the positive data into the subset 2 box. Now that we have the data for our two subsets, click the Advanced Workflow tab. Click the Analysis button and a drop-down will appear. Click on Hierarchical Clustering. We now need to define the variable for our heat map. To define the variable, we'll need to drag in high-dimensional data. To find the high-dimensional data, go back to the Breast Cancer Study by Sorley folder and expand the Biomarker Data folder. Expand the Gene Expression folder and expand the Stanford cDNA Microarrays folder. Now we see data with the DNA symbol listed to its left. We know that we have found high-dimensional data when we see data that is listed with the DNA symbol to its left. Drag in the high-dimensional data into the variable box, and be sure to click the High-Dimensional Data button. If you do not click this button, your analysis will not work. When you click the button, a Compare Subsets Pathway selection pop-up will appear. Make selections in this pop-up that will meet your analysis needs. For this example, I am using the default settings. Click the Apply Selections button. When the pop-up closes, scroll to the bottom of the page and click the Run button. The analysis will take a moment to process. When the analysis is completed, scroll down in the window to view your heat map. Click on the heat map, and an image will open in a new window for improved readability. In the Hierarchical Clustering heat map, we see that data is organized so that objects in the same cluster are more similar to each other than to those in other clusters. Subsets are marked along the x-axis on the top edge, and genes or probes are listed on the y-axis or the right edge. Back in the tranSMART window, click the Save to PDF button to save this hierarchical clustering. In the instance of tranSMART, click on the Dataset Explorer tab. K-means clustering is a visualization of groupings of the most closely related data points based on the number of groupings you specify. With K-means analysis, only columns are clustered together. Rows are not clustered. To get started, expand the Public Studies folder, and we'll be using the Breast Cancer Study by Sorley data. Expand the main folder, expand the Sample Factors folder, and expand the T&M category folder, as well as the Tumor Stages folder. I'm going to click and drag the T3 data into the subset 1 box. I will also be doing a subset 2 for comparison in this K-means clustering heat map. To get my second set of data, I will open the estrogen receptor assay folder, and I will click and drag the positive data into the subset 2 box. Now click the Advanced Workflow tab. Click the Analysis button, and from the drop-down, select K-means clustering. We now need to define the variable for the heat map. To do this, we need to find high-dimensional data. Go back to the Breast Cancer Study by Sorley and expand the Biomarker Data folder, expand the Gene Expression folder, and the Stanford cDNA microarrays folder. And we see data with the DNA label to its left. This is our high-dimensional data. Drag the high-dimensional data into the high-dimensional data box, and be sure to click the high-dimensional data button to ensure that your analysis will work correctly. When you click the button, a Compare Subsets Pathway Selection pop-up will appear. Make selections that suit the needs of your analysis. For this example, I will be using the default settings. When you are happy with your selections, click the Apply Selections button. Now scroll down. At this point, you can change the number of clusters you want for your k-means clustering heat map. I will stay with the default setting, 2. At the bottom of the page, click the Run button. The analysis will take a moment to process. When the analysis has completed, scroll down on the page and you will see the k-means clustering heat map. Click on the image and it will appear in a new window for better readability. Columns that fall into the same cluster are represented by the different colors on the top bar of the heat map along the x-axis. Genes and probes appear along the right edge, and subsets are grouped along the bottom edge. In a k-means clustering heat map, genes or samples are clustered into a specific number, in our case, 2. Each cluster is centered along a randomly selected data point. Back in the tranSMART window, click the Save to PDF button in the right-hand corner if you wish to save this heat map. In the instance of tranSMART, click on the Dataset Explorer tab. Marker selection is a visualization of differently expressed genes in distinct phenotypes. The algorithm determines the set of genes that is most differentially expressed between two subsets. We will be able to view a list of differentially expressed genes presented in a table with accompanying statistics. Marker selection heat maps require 2 subsets. Expand the Public Studies folder. We will be working with the breast cancer study by Soily. Expand that folder and expand the sample factors folder and expand the TNM category folder and finally expand the tumor stages folder. I'm going to click and drag the T3 data into the subset 1 box. For subset 2, I will expand the estrogen receptor assay folder and drag the positive data into the subset 2 box. Click the Advanced Workflow tab. Click the Analysis button and click on Marker Selection. We now need to set the variable for our marker selection heat map. We need high-dimensional data to do this. To find the high-dimensional data, go back to the breast cancer study by Soily and expand the biomarker data folder, the gene expression data folder, the Stanford cDNA microarrays folder, and there we see data that has a DNA symbol listed to its left. High-dimensional data is denoted with a DNA symbol to its left. Drag the high-dimensional data into the box and be sure to click the high-dimensional data button to ensure that your analysis will work correctly. A Compare Subsets Pathway Selection pop-up will appear. Make selections that will suit the needs of your analysis. I am using the default settings for this example. Click the Apply Selections button. Under the high-dimensional data box, there is a field labeled Number of Markers. This number can be changed based on the needs of your analysis. For this example, we will stay with the default 50. Scroll to the bottom of the page and click the Run button. The analysis is being processed. This will take a few moments. When the analysis has completed, The marker selection heat map will appear, and you will view it by scrolling down on the page. Click on the image of the heat map to open it in a new window. We see a display of differential gene expression. Individual values are represented by different colors. Go back to the tranSMART window. At the bottom of the page, we see the top markers table. This table represents the user-specified numbers of differentially expressed genes between the datasets. Sort the table by clicking on the column headers. Save a copy of the marker selection heat map and of the table by clicking the Save to PDF button in the top right corner. You have now seen all of the heat maps that tranSMART allows users to generate. For more assistance with heat maps, see the user manual at tranSMARTfoundation.org.